This is being brought to you courtesy of the ADRC Biomarker Core, in particular the subset of us that work on fluid biomarkers, and Paula and I are the co-leads. As many of you know, I'm an enthusiast about biomarkers. I think that they can help us tell a lot about what's going on in the brain. Just to put a few things in perspective, some of the ideas that are percolating around Alzheimer's and its staging, a number of folks led by Cliff Jack, and then this has had a little bit of updating, have proposed a research framework to define a biological definition of Alzheimer's, the notion being that one needs amyloid pathology as well as tau pathology and some component of neurodegeneration to actually define Alzheimer's disease. Amyloid and tau alone constitute Alzheimer's pathological change, and cognitive impairment may occur anywhere along the whole spectrum. But in fact, if we look at autopsy studies, we find that there are many, many people whose brains harbor other pathologies, sometimes on their own, sometimes in varying combinations. The big ones are alpha-synuclein pathology, TDP-43 pathology, which has also been called late, and then vascular pathology, and these may all contribute to the clinical pathological relationships. So there's been a lot of research on trying to bring these the ATN framework to life, and regarding plaques and tangles. For amyloid, we can do a PET scan or measure CSF or plasma A-beta 42/40. For T, we can do a tau PET scan, and then there are some plasma p-tau markers that relate to tau, although as it turns out, they relate more strongly to amyloid PET for the most part. Neurodegeneration, one can think of all sorts of things that might be associated with eventual brain atrophy and neuron loss. There are anatomical perspectives by doing an MRI to look at atrophy or pathways within the brain that are undergoing neurodegeneration. PET scan, FDG glucose PET is one example. Biochemistry, some measures of tau may reflect neurodegeneration. Neurofilament light is one marker, and there are a host of others. So if we're going to go after biomarkers in fluids, there are a host of different things that can be measured, and I've listed almost all of them over here. Proteins have received the most attention, but within proteins, we can measure the whole protein, post-translational modifications, which turn out to be quite important in Alzheimer's disease. And I've listed some of the options here: truncation, phosphorylation, nitrosylation, etc. We can try to measure aggregates or seeds. Metabolites, we can do metabolomics, which reflect all sorts of metabolic pathways. For lipids, there are a host. There are over 70,000 different kinds of lipids that can be measured. Usually these are lumped into different classes of lipids, and we can either measure free lipids, or we can try and isolate lipid particles from fluids and see what their constituents are. We can look at cell-free RNA. And so there are all sorts of different RNA fragments, messenger RNA, microRNA transfer, and piRNA are some of the main ones that have been characterized. Cell-free DNA, we can look for DNA fragments or for epigenetic markers on the DNA fragments to try to tell us something about the cells of origin or a little bit about the biology. Extracellular vesicles, which would include exosomes and ectosomes. And then cells, and we've been involved here with others in isolating T cells from CSF. We can also look at T cells in the periphery, for example. What samples can we use? So CSF has received a lot of attention. Lumbar CSF gives us some fairly direct insights into the brain, although it differs from interstitial fluid, which might be a bit closer to what's going on within neurons. CSF is a little bit invasive, and it may be harder to sample longitudinally than blood. So blood, we can readily get plasma or serum from people. It is a less direct reflection of the brain. And the most informative markers in the blood are things that we can measure that we think originate from the brain, which are likely to reflect CNS disease, rather than the majority of markers that can be measured in blood that occur in the periphery. Urine has been looked at. There are a host of issues and questions, namely variable production rates of urine, extreme dilution of markers by the time they've started in the brain, perhaps got into the blood and then got into the urine. Skin biopsies have been used for histology or some other approaches of analysis. I omitted saliva. There are a few investigators that are looking at that as well. So how are we going to use biomarkers for Alzheimer's and related disorder? There's clinical use for diagnosis, prognosis, risk, monitoring, and so forth. We can use these as an adjunct to clinical trials or treatment studies. One important use is to assess whether the intervention results in target engagement. It is also possible to use some biomarkers as outcome measures in clinical trials. And then most biomarkers, at least at an early stage, are studied to try and improve our understanding of disease. For example, the course of disease, staging, different biological processes, and I've listed some of these which have been looked at in great detail. For example, inflammation, neurodegeneration, endolysosomal function, and so forth. Improve understanding of how genetic risk might be mediated, and then look at other risk factors, for example, age, sex, or environment, and ask about how they translate into biomarkers and potentially into mechanisms. If we're going to use biomarkers with a capital B, and this is for approval studies that the FDA might consider, and these would get into regular clinical use, the FDA came up with a framework for qualifying biomarkers in 2018 called Biomarkers, Endpoints, and Other Tools, or BEST. And the assumption is that you have a biomarker where it's underwent analytical validation that's been very good, and now we are trying to figure out how are we going to validate this clinically. And this is actually important for writing grants and trying to propose how to study biomarkers, because we need to think about the context of use of a biomarker. And here are a number of examples. I won't go through all of them, but clearly they all have definitions. And in thinking about a biomarker that you might want to measure, it may be useful to run through this framework and try to figure out which aspects the biomarker might potentially apply to. So I'm going to focus a lot on proteins, and I'm going to start off with how we actually start by measuring proteins with the hope of developing them into biomarkers. So if we have a candidate biomarker and we want to measure it, we can set up an assay. The most commonly used and widely available are ELISAs. And in some instances where these are translating to clinical use, there are fully automated ELISAs that have been developed. These have a much lower degree of variability because steps of washing and pipetting are taken out of the hands of technicians, and the machine does this with much less variability. In the last decade or so, we've seen ultra-sensitive assays capable of detecting very low picomolar or picogram per mil levels of analytes. An example is a generic term of SMOA, or single molecule analysis. There are a couple of devices that do this. And then mass spectrometry is widely used to assess candidate biomarkers, provided calibrators for those markers are used. The other approach is to do omics or discovery for biomarkers, and there are a number of different approaches. These are some that are used most commonly. Mass spec is very widely used, and there are a host of different approaches. I won't go into all of the details. We can perhaps discuss these at later meetings. There are top-down and bottom-up approaches, and what's become very popular in recent years is to do what's called data-independent acquisition, where one gets thousands of different spectra from the sample of interest and then maps these onto databases to be able to name peptides and proteins and then study these. It's possible to put together a panel of proteomic markers to measure by mass spec, and some of the methods that are used include SRM or TMT, where in these calibrators are used, and specific labels for proteins that can measure up to about 100 to sometimes 150 proteomic markers quantitatively. Immunoassays have been developed in different ways, and one of the ways to get a readout is instead of doing a standard ELISA with two antibodies in a sandwich, one can use a secondary antibody that is labeled to an oligonucleotide sequence, and PCR is done to amplify that specific sequence. One of the platforms that's very widely used has been developed by a company called Olink, and Olink has developed a host of different assays. They have currently more than 4,000 proteins that can be measured in approximately 20 to 40 microliters of starting sample, and they have a lot of data that's been generated on plasma and CSF. Another approach uses aptamers. Aptamers are kind of sequences of DNA that fold in certain directions and can bind proteins. And by having very large aptamer panels, one can come up with specific aptamers that bind to proteins selectively. A company called SomaLogic has specialized in doing this, and they have very sensitive, somewhat quantitative assays. Their most recent panel can measure 11,000 proteins, needing 55 microliters of fluid. So lots of ways to do omics, and these are somewhat complementary to one another. They don't overlap completely, and they don't produce exactly correlative data. So if we're going to take an assay and try to develop and validate it and try then to use it at scale, we need to do a few different things. The first is the method of analysis or verification. And so this requires, typically for an ELISA, defining parameters such as how specific is it for the particular protein or peptide we're going to measure, what is the lower limit of detectability, the lower limit of quantification, what is the dynamic range, over what range is the assay linear, and what is the upper limit of quantification. We need to define the reagents that are going to be used. This will include the antibodies, also calibrators, and for some particular assays, especially if they get into a clinical arena, standard reference material. So for Alzheimer's disease, an example of SRM is that huge pools of CSF have been prepared that have high, medium, and low levels of A-beta 42, ascertained by mass spec to define a standard reference material for calibrating CSF A-beta 42. One needs to then run the assay on different occasions and define test, retest, coefficient of variability, repeatability, and precision of the assay. Once you have all of this data pinned down, the next would be to do an initial analytical validation and qualification. And so the usual thing that is done is to compare cases and controls against a gold standard. Accuracy, reproducibility, is the assay fit for its intended purpose? And one needs to calibrate how this, or document how the samples were actually prepared and handled, and whether this actually makes a difference to the assay. Having done all of these steps, the next is to do a wider clinical validation. So again, this defines context of use, the specified patient population, a rigorously designed study of one or more validation cohorts or populations, and then determine accuracy, specificity, reproducibility, and how robust is the assay, and then parameters like sensitivity, specificity, positive and negative predictive value. So all of those would be things that we would want to do if we're going to get an assay all the way to clinical use, or at least to very widespread research use. Clearly, some assays don't go through all of this if we're just interested in more limited discovery. So I'm going to talk a little about CSF biomarkers related to amyloid and tau, because these are among the workhorses that we use in the AD field. First off, CSF A beta In the cartoon on the left, CSF-A beta results from the serial cleavage of APP, the parent protein, and there are a number of different secretases, alpha and beta being the most upstream ones. These cleave the large N-terminal fragment and leave a C-terminal fragment of APP, and then another molecular scissors called gamma secretase comes along, and it cuts this peptide sequence within a membrane at a bunch of different amino acids, and it produces peptides ending at amino acids 37, 38, 40, 42, and actually I left out 43. And all of these can be measured individually. A beta 42 turns out to be the amino acid form of A beta that has the highest propensity to aggregate, and it is typically the one that forms the majority of amyloid plaques. In CSF, we do not measure full-length tau. This is a cartoon of one of the forms of full-length tau. There actually are 6 different isoforms. And a number of proteomic studies have been done to isolate fragments of tau that could be located in CSF, and this gives you an idea of how complicated things are. These are different widely used tau antibodies against particular epitopes that have been used. So it turns out that most of these tau fragments either are somewhere towards the N-terminal of tau, and we can measure tau, a number of these different fragments using different sorts of assays. Phosphotau can be measured specifically as well, and phosphotau can be used as a marker for tangles, and there are a number of fragments. They don't go usually beyond roughly this length of tau. So there are a number of fragments that can be IP'd, and then one can develop assays for these. So let's go through a little bit of the data. In Alzheimer's disease, the finding of a low CSF-A beta 42, or one can try to make a ratio of 42/40. This is a marker that amyloid has been deposited in fibrillar aggregates within the brain. And the other way to detect these aggregates is through amyloid PET. And so there have been a number of studies. This is an example from ADNI, showing that if one looks at amyloid PET on the horizontal axis, here's a cutoff where people would be PET positive in this zone over here. And if they're PET positive, then the A beta 42 is almost always low. If they're PET negative, the A beta 42 is usually high. You can see that there are a bunch of people here where the amyloid PET tends to be, there's a bit of a mismatch in some cases, slightly more often for a low A beta 42, which may drop a little before the threshold of detecting amyloid PET through different ligands. If you do a ratio of 42/40, it tends to put a few more cases into either of the blue boxes, and it may give a slightly better overall classification. So if we look at the relationships between amyloid PET and tau PET and cognitive decline, this is turning to PET studies, one can find that there is a relationship. So PIB is a biomarker for amyloid PET, and in general, anterior temporal and posterior medial deposition of tau, which can be measured by doing tau PET, are associated with people who are more likely to be amyloid positive. And this will predict people who are more likely to show memory change, which was looked at in a number of different approaches in this particular study. So again, we might want to try and look at when do CSF biomarkers change according to the amyloid burden, and does this predict cognition? So to go back to amyloid PET scanning, there's now a method to try to standardize amyloid PET across different ligands, and a term of centiloids has been developed. People who are amyloid positive, regardless of the ligand, tend to be at a threshold of roughly about 20% of the centiloid threshold. And so if one looks at, this is a cognitively unimpaired cohort, these are different definitions of thresholds of centiloid positivity. And this is the percentage of people who had abnormalities of phospho-tau, total tau, neurofilament light, and neurogranin, which is a synaptic marker. And you can see that a percentage of people are showing abnormalities in these markers, even at this incredibly early change. So these may be markers that one might think of to look for as signs of neurodegeneration, which might be beginning even when the amyloid burden is very low. I mentioned that there are fully automated assays, and one example of this is an assay platform developed by Roche called Elixis. There's another one called Lumipulse, developed by Fujirebio. These are fully automated assays for CSF. The Roche one does A-beta 42 tau and p-tau. The Elixis one does all of these as well as A-beta 40. And this came from data from two different cohorts where the gold standard was amyloid PET. And the idea was amyloid PET is a gold standard for amyloid positivity. It's been validated against autopsy. How well do these fully automated assays do? And if we use different combinations of markers, how well do they do? So in this study, taking a ratio of phospho-tau 181 over A-beta 42 had a very good AUC and ROC curve of 0.94. Total tau did almost as well. Individual biomarkers did well, but not quite as well. And cutoffs were proposed. And in fact, this assay has received recent FDA approval and has been implemented by the Mayo Clinic. There is similar data using the beta 42 over 40 ratio from Lumipulse, and that data is also FDA approved. So those are amyloid and tau biomarkers. If we're going to think about neurodegeneration, it's really a very complicated entity to think about. There are many potential protein biomarkers, many potential biological processes. So if we think that the drivers of neurodegeneration are A-beta pathology, and a number of markers we could use for that, and then tau pathology and a couple of different markers, there are a host of different changes that could happen, maybe simultaneously, maybe some are earlier, some are later. Inflammation involving microglial and astrocytic activation can be part of the neurodegeneration process, and there are some markers that can be measured as listed here. Synaptic function, damage to synapses, or loss of function of synapses can be part of neurodegeneration. Neuronal injury, release of markers such as neurofilament light. There are a couple of others listed here, VILIP1 and fatty acid binding protein 3. Alpha-synuclein is a synaptic-related protein, and it could be released or caught up in some aspects of neurodegeneration. And then vascular and blood-brain barrier dysregulation might play a role in neurodegeneration. So lots of different things that could be measured. I'm going to look at a bit of data on one of them, which is neurofilament light, which has been very widely studied. This is an analysis across a number of different biomarker studies looking at what happens with aging. Neurofilament light in CSF increases with aging. You can see there's quite a dramatic increase actually from age 20 projected all the way to age 100. So in fact, if you're going to look at a particular disease, you may need to actually standardize for age. And that's the trajectory among people who are neurologically and cognitively normal. In Alzheimer's disease, it is increased. I think this is CSF and this is plasma. Looking at the two different. Oh no, I'm sorry. This is Parkinsonian disorders, and this was dementia disorders. So here are some of the trajectories. And here is Alzheimer's disease, so it's a small increase. Frontotemporal dementia, there's a greater increase. Dementia with Lewy bodies overlaps with AD and so forth. And here are some Parkinsonian disorders. Parkinson's disease doesn't, you need a magnifying glass to detect the very small changes. Maybe a little bit more if people have Parkinson's with dementia, maybe because of coexisting Alzheimer's disease in a number of cases. In certain movement disorders, atypical ones, PSP, progressive supranuclear palsy, and multisystem atrophy, it turns out that an increase in neurofilament light can be found. So it's a non-specific marker, but it can be useful in helping with diagnosis or perhaps longitudinal assessment of some of these markers. So I've talked a lot about CSF testing. Can we detect a number of these markers in blood, and are they useful? And the answer is yes. Let's talk about, firstly, looking at amyloid beta protein markers. So these are data from what has come out as so far the most robust test for plasma A beta. It's done by immunoprecipitation and mass spectrometry by a company called C2N. They've named their test Precivity. And these are data from one cohort study where the ratio of 42/40 was lower in people who are amyloid positive than amyloid negative. You can see that there's quite a lot of overlap. And over here, they came up with a probability score where they looked at the numbers, and they also included age and ApoE genotype. And by throwing those all together into a composite score, they're able to get better classification with still a moderate degree of overlap, certainly CSF overlap than this. This is a study that looked at combining plasma 42 and 40 and tau and neurofilament light and ApoE, and all of those gave a moderate ROC curve, probably comparable to 42/40, and this was published in 2019. Since then, we've seen a lot of work on measuring tau fragments in plasma. The best of these so far has been p-tau 217. I'm only going to show you one paper. There are probably several hundred at this stage on p-tau 217. A number of different assays have been developed, sometimes using some of the ultrasensitive assay methods. This is a study where amyloid biomarkers, either CSF or PET, were used to divide groups. These were either cognitively unimpaired or MCI who converted to AD or didn't convert to AD, and divided up by amyloid positive in blue or amyloid negative in green. So again, the actual levels of this protein are extremely low, less than 5 nanograms per liter or picograms per mil. But over time, with 5 years of follow-up, you can see that if you're amyloid positive, there is a group increase, whether you are cognitively normal, whether you have MCI, the baseline starts higher and there is an increase. And MCI, if you converted to Alzheimer's, looks pretty good. If you didn't convert to Alzheimer's, it doesn't really increase over time. This shows neurofilament light in the same cohort, so no real signal in cognitively unimpaired. If you're MCI, there's a very small signal with quite a lot of overlap. If you converted to Alzheimer's, again, a small signal with a lot of overlap, certainly 217 beat MCI. People have tried to figure out, can we work this into a clinical workflow? If we were going to use some screening cognitive tests and plasma p-tau, could we come up with an algorithm? And this was done using data from a Swedish study called Biofinder and ADNI, and they threw in a bunch of different variables for models, and their best predictive model included p-tau 217, ApoE genotype, memory and executive function testing. You could gain a little bit more by throwing MRI and a little bit more, by throwing in one more marker, but really this was pretty good. If you just did plasma P30 and cognition alone, but you had to have a good test of memory executive function, the area under the curve was 0.89, so pretty good, and they came up with a slider to try and do this. So this could be used at a memory disorders clinic, where memory and executive function could be tested fairly rigorously, but would not yet be appropriate for a primary care clinic. So I've shown you longitudinal data. Some of the longitudinal data has been used to try and line up biomarkers. If we were going to use biomarkers to screen people and to try and do prevention trials for Alzheimer's, for example, here is a schema of when things change and how many years before the likelihood of detecting, for example, neurodegeneration might you start, and what could the marker be and what could the target be. So the earliest markers, which you could see even 20 years before onset, would be amyloid biomarkers. If you're looking at hypometabolism, for example, PET, atrophy, maybe some neurodegeneration markers. You could maybe start 5 to 10. And tau aggregation would be closer to symptomatic disease and more likely to be used in a secondary prevention trial. So to show you that this can actually be achieved, there are a couple of clinical trials that have now published data in which they've looked at treatment with anti-amyloid antibodies and CSF or plasma biomarkers. This was a paper from the Diane TU trial. This was a clinical trial in people who have autosomal dominant Alzheimer's disease. There were two different arms of this trial. In one group, people were treated with an antibody that can clear amyloid. This is shown over here. And this was gantenerumab. In another arm, they were treated with solanezumab, which does not clear amyloid. And remarkably, in the study, one can see that in the Gant-treated group, a number of different markers change relative to placebo. This includes markers of amyloid deposition, which hopefully would happen. And they were able to show that CSF tau and p-tau decreased as well, neurofilament light to a lesser degree. These actually happened in the absence of showing a cognitive benefit, but at any rate, there was an important biomarker signal in two of the other anti-amyloid trials. They have also shown improvement in plasma biomarkers in MCI or mild AD. How about vascular change, which I mentioned is something that can contribute to memory changes? It's been really hard to come up with convincing fluid biomarkers for vascular dementia. One of the gold standards for vascular cognitive impairment is analyzing T2 white matter MRI burden. And this was a study looking at a host of different CSF endothelial or inflammatory markers and lining them up against burden. There's a marker called placental growth factor that has to do with endothelial function to some degree in the brain. It clearly originally came out of placental research, so whatever it does in the brain was not well known at the time. And they did find a relationship with T2 white matter burden that was independent of amyloid burden in the study. So this might be a marker that warrants further study. Otherwise, nothing clear that we can measure in biofluids. One other marker to discuss is alpha-synuclein. So this is going to be a little different than the markers I've shown so far. All of the markers we've been interested in somehow relate to phenomena where proteins, A beta or tau, have aggregated, and then this causes brain pathology downstream. Alpha-synuclein does the same thing. It forms aggregates that can form Lewy bodies or Lewy neurites and can trigger disorders like Parkinson's disease or dementia with Lewy bodies or multisystem atrophy. So what is revolutionizing our approach to these disorders is the ability to detect seeds of alpha-synuclein. And so this is an assay called a seeding assay. The idea is that one takes the sample, which might contain some small number of pathogenic seeds, put it into a microplate, and add recombinant alpha-synuclein as a fuel. One then shakes the microplate for about 5 minutes, then you let it rest for 5 or 10 minutes, and then you keep doing this over and over again. The normal synuclein bumps into the seeds, and at some point, the seeds corrupt normal synuclein and makes them form protofibrils. These can disaggregate and keep seeding. One adds a bit of thioflavin, which can detect amyloid configurations and measure fluorescence. And lo and behold, over time, a large fluorescent readout occurs if there were seeds. Here are data from some of the earlier studies in this area, and there are now at least 100 from some very, very large cohorts. This shows the difference between CSF from Parkinson patients, where you do get seeding. In this case, it took 200 hours or more, versus normal controls. If you immunodeplete the seeds from Parkinson's CSF, we don't have perfect antibodies to do this, but it reduced the seeding compared to the same CSF sample. And this was a study of PD patients versus controls with quite an impressive difference. Here was a larger study looking at a host of different synucleinopathies versus controls, Alzheimer's, PSP, MSA. This is a study trying to do the same thing in skin biopsies, where one can achieve seeding with some good initial results. And there are efforts to detect these seeds in plasma that are ongoing. I'm not going to show you much more data about this. Again, this could be a topic we could have an enormous session all about. I wanted to touch on the question of what biological events are fluid biomarkers measuring. So we can measure all sorts of things, and the question is, what are they telling us? So we can measure events related to A-beta deposition and aggregation, and this is what we think the A-beta 42/40 ratio relates to. When we measure these different tau fragments, we think they have to do with tau cleavage and phosphorylation. If we think about damage to neurons and axons, this may be how neurofilament light and some of the synaptic markers get into brain. There are some other markers released by synapses, and as synapses are lost, these are actually decreased in CSF. There's a marker called neuronal pentraxin 2 and another one called VGF that are decreased in Alzheimer's and in some other neurodegenerative disorders. We have a couple of markers that are related to microglial activation. One of them is a secreted form of TREM2 detectable in CSF. For astrocytes, one can detect glial fibrillary acidic protein, GFAP, in both plasma and CSF. Senescence is a topic of great interest. It's not clear that we have a great marker. Some people have claimed IL-6 levels may be changed in certain tissues. And then the blood-brain barrier is of great interest. We don't have great markers, but excuse me, measuring the CSF to plasma albumin ratio is one approach. So how about combining? We've talked about single markers and maybe a few of them all together. How about the option of combining systems biology and omics? If we measure hundreds or thousands of biomarkers in CSF or plasma, how do we determine what biological processes these may be telling us? And so there are a number of approaches to do this. Again, we can have some complete later sessions about this. It's an increasingly interesting topic. It's been used to study Alzheimer brain samples as well as CSF, and the usual approach has been to do mass spec, somewhat quantitative proteomics, usually using label-free or DIA mass spec, and then doing an unbiased network analysis, trying to decide out of all these peptides and proteins, what are the cell types of origin, what are the biological pathways and ontologies. One can try and map these onto genetics, or you can look at protein-protein interactions. And some of these statistical methods cluster proteins into modules. In Alzheimer's, for example, it's possible to come up with a host of different modules, something like 20 in the brain and about 12 in CSF. Some of the ones that are illustrated in this picture show elements of inflammation with some cell types of origin that can be detectable in the brain and CSF and are increased in Alzheimer's. Some myelination markers related to oligodendrocytes, and again, these are increased. RNA binding and splicing, not any particular cell type in specific. Again, these are increased. Some markers tend to be decreased, synaptic markers. If you develop a whole panel, some of these cluster into a decreased panel or module, mitochondrial markers, energy metabolism, and cytoskeletal markers. And one can cluster these together and use integrative omic approaches. I could be happy to share some papers, but not enough time to go into depth right now, looking at local proteomics, either of brain samples or biofluid samples, do all of this clustering, network analyses, and try and integrate and come up with biomarker prioritization. And some of the prioritization issues could be, is this a biological process that makes sense in terms of the disease? Is this reproducible? How big is the magnitude of effect? Do we get agreement across a bunch of different cohorts and so forth? And then one can come up with more targeted biomarker assays. Instead of measuring 4,000 markers, let's take the top 120. And again, you could use mass spec methods in which you don't require antibodies. And you can come up with multiplex assays, and then immunoassays, which are usually of more widespread and easier use, and come up with the same sort of approach. And eventually, the hope would be to come up with network-based clinical biomarker assays. So an important and emerging approach, and we'll talk more about this in future. So in the remaining time, I wanted to talk about resources. And I'm going to start with resources that are available at the UCSD ADRC. So we have banked plasma, and this has been done from all ADRC participants at baseline and every 2 years, starting in 1990 through 2020. And then in 2020, we started doing this annually. This covers an age range of more or less 60 to 101. There are mostly white participants. About 200 plasma samples so far come from Latinos, or rather 200 subjects, actually. There can be multiple samples. The baseline diagnoses are normal MCI, AD dementia, which can be mild or moderate, starting off. We have some samples from people with frontotemporal dementia and some with DLB. And of our database, we have submitted 315 plasma samples so far, which have been analyzed for p-tau 181, 42 over 40, and neurofilament light of people seen in the last 3 years. We prepare a Buffy coat, and some of this is used for DNA extraction from everybody at baseline. We do have some Buffy coat residual, which some people have used to extract cells. Our CSF bank consists of CSF from approximately, it's a bit more than the 600 participants. It's done at baseline, and we are trying to get longitudinal CSF samples from participants more or less every 2 to 3 years. We have it from about 50 samples so far. And these samples are of particular use once we start analyze them in the context of very deep phenotyping. So we have clinical data of all different kinds, neurocognitive data, medications, longitudinal data on everybody, apoegenotype on everybody, GWAS level data on at least 80%, research grade MRIs on about 250, amyloid PET on some, about 60 or 70 to date, tau PET about 20. In addition, I've been collecting CSF from the Memory Disorders Clinic more recently, and we have, it's a little over 30 CSF samples so far from clinic patients with MCI or AD that have been banked for research, and this number is going to grow. There are lots of other biomarker sample and data resources, and I'm only going to list a few of them here. One of them is ADNI, the Alzheimer's Disease Imaging Initiative. ADNI is a multi-center longitudinal study collecting data and MRI on cognitively normal people, MCI and AD from, I think it's 54 different sites in the US. Started in 2003, there were more than 1,200 participants. Everyone donates plasma. They've had CSF on about 40% and some longitudinal CSF. They've run a lot of assays already, and these can be downloaded from their database. They have done, I mentioned SomaLogic as being one of the omic type analyses. They've done SomaScan on, I think, 1,000 CSF samples, and I think they've run the 4,000 analyte panel. They have some mass spec metabolomics, mainly on plasma, some CSF, and some targeted proteomics. Genetics, there's ApoE genotyping and GWAS on everybody, and more recently, whole genome sequencing on the entire cohort. Over 500 have had amyloid PET scans, more than 200 have had tau PET. So a very useful database, lots of publications already. NAC and NICRAD, these are large databases and biosample repository resources that serve all of the different ADRCs across the country, as well as some other studies. These collect standardized clinical and autopsy data, which all goes into NAC, from the network of more than 30 ADRCs. It includes people with normal cognition, MCI, AD, and some non-AD cases. There are at least 30,000 subjects with clinical data. There are longitudinal data, lots of autopsy data. There's DNA, GWAS, some whole exome, and some whole genome sequencing data. There's some MRI data on maybe a couple of thousand people, and they're starting to collect amyloid and tau PET data from the centers. Plasma is banked, and again, the same set that we have run, or we have had run, is run by the same person at NICRAD on these biomarkers in plasma over the last 3 years on probably a few thousand people by now. They have blood pellets and immortalized lymphocytes, and you can put in a request for data and also for biosamples. Some other resources to mention. This is more along the alpha-synuclein spectrum. PPMI, funded by Michael J. Fox, is a longitudinal Parkinson's cohort. They also include people with REM sleep behavior disorder, people who are genetically at risk for Parkinson's, and initially 200 controls, with more controls being recruited. Overall, more than 1,500 participants and rising. People have annual CSF for at least 3 or 4 years. All of these different blood products, cell lines through blood draw, not through skin biopsy, and DAT scan to assess dopaminergic neurotransmission function. And CSF has so far been analyzed for AD biomarkers and for alpha-synuclein seeding amplification assays, and some other markers. PDBP is short for the Parkinson's Disease Biomarker Program, which is funded by NINDS and managed by them. And they have funded a series of UO1 grants that have recruited cohorts of Parkinson's, usually mild to moderate, some controls that match these cohorts, and there have been 3 or 4 cohorts in the multi-center study that we're part of dementia with Lewy bodies. All of these patients have some standardized clinical phenotyping. There's limited longitudinal data. Usually there is either cross-sectional or maybe 1 year of follow-up in most of these cohorts. What is available is DNA, plasma, CSF on some of the cohorts, MRI, DAT scan, and PET on some as well. Also like to mention some consortia that have been developed that are doing extraordinarily large-scale work nationally and internationally. AMP-AD, Accelerated Medications Partnership for AD, has a number of different sponsors, including NIH. And this includes the multi-omic characterization of post-mortem brain tissue from AD, non-AD, and controls. They've done RNA-seq, some single-cell RNA-seq, epigenetics, protein and metabolites, and lots of publications. Plasma and CSF is in progress, and there is some proteomic mass spec data available. The Allen Institute has a host of different omic analyses of all sorts of species in progress regarding humans and Alzheimer's disease. There's a project called the Allen Brain Cell Atlas, which has achieved multimodal single-cell data across the mammalian brain in relation to aging. They have some childhood cases as well. And then there's also the Alzheimer's Disease Brain Cell Atlas, which is done in conjunction with the ADRC at University of Washington. And again, this has done multi-omic data analysis, including some single-cell analysis, and it's an extraordinarily detailed atlas. One other one to mention, this is a work in progress. This is the Global Neurologic Proteomics Consortium with a host of different funders, and they have used primarily SomaLogic and to some extent Olink data. And this is what has come in out of their initial wave from 17 different cohorts, 9,000 Alzheimer's samples, bunch of ALS, ton of healthy aging, 2,000 P.D, mostly from PPMI, FTD samples from a study called All FTD. And they have been trying to harmonize the data. Anybody who's interested can contact them and find out where things stand or whether it's possible to download certain data sets. They have a listing of committed cohorts. They also have a listing of what they call engaged cohorts. And we are potentially one of the engaged cohorts. It might be possible for all of our ADRC plasma and CSF samples to undergo analysis with SomaLogic to be part of this consortium. So I'll stop there.